right, and now for the moment you've all been waiting for, time for us to dive into today's sessions. For awareness, we will have two initial presentations followed by a Q&A with our first two speakers, then an additional presentation followed by a Q&A with all presenters and panelists. So now it's my pleasure to introduce our speakers. First, we'll hear from Helen Heyman, Regulatory Health Project Manager um, with the Executive Program and Project Management staff in the Office of Clinical Pharmacology within the Office of Translational Sciences within the Center for Drug Evaluation and Research. Then we'll hear from Dr. Xining Jiang, a policy lead with the Guidance and Policy team, also with the Office of Clinical Pharmacology and Office of Translational Sciences within CDER. So please join me in welcoming our first presenter, Helen Heyman.
Welcome to today's SBIA webinar on the ICH M12 drug-drug interaction studies final guidance. It is a great pleasure to be here today. My name is Helen Heyman. I work in the Office of Clinical Pharmacology as a Regulatory Health Project Manager. In terms of webinar objectives, I will start by giving an introduction on the framework of this guidance. This will be followed by a presentation by Dr. Yang on in vitro DDI assessments with a Q&A panel. Dr. Reynolds will then cover clinical DDI assessments. There will also be time for questions at the end of her presentation. In my introduction, I will cover the rationale for the ICH Drug Interaction Studies guidance, its goal and scope, process and timeline, and finally implication of adoption by the FDA. Now let's talk about the rationale for the ICH Drug-Drug Interaction Studies guideline. First, why focus on drug-drug interactions? As you know, drug-drug interactions, or DDIs, may occur when patients are taking more than one drug. Some patients, in particular fragile older patients or patients with serious or multiple health issues, can be prescribed a large number of different drugs. Therefore, it is important to consider an investigational drug's potential to interact with other drugs and to decrease the risk of adverse events or loss of efficacy when patients are taking more than one drug. Given that it is impractical to evaluate every DDI in clinical trials during product development, systematic risk-based approaches are used to assess DDI's potential. So why globally harmonize? While regional guidelines for investigations of DDIs using risk-based strategies have been available for decades to assist drug developers, some differences remained among regional regulatory guidelines, leading to inconsistent expectations and recommendations. The impact of these inconsistencies include different clinical interpretations and translations across regulatory agencies. The International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, ICH, a convener for harmonization between regions, enabled the development of the M12 guideline through strong alignment across regions to harmonize stepwise risk-based strategies used by multiple regulatory agencies to assess DDIs. Now a few words on the ICH. The ICH promotes public health and helps reduce the uncertainty for the pharmaceutical industry to meet the requirements of multiple regulatory agencies, thereby leading to more efficient use of resources, reducing drug development costs, and making treatments available to patients earlier. Let's now talk about the goal and scope of the ICH M12 drug interaction studies final guideline that was approved in May 2024. The guideline's goal is to harmonize recommendations that promote a consistent approach for designing, conducting, and interpreting in vitro and clinical DDI studies during the development of a therapeutic product. In terms of scope, it is the first globally harmonized regulatory guideline on assessments of pharmacokinetic drug interactions mediated by metabolic enzymes and drug transporters. In this table, you can see in and out of scope topics. For example, the guideline covers small molecules and biologics such as monoclonal antibodies. The guideline does not cover new modalities such oligonucleotides. Now let's turn to the process and timeline for the development of this guideline. This will give you an idea what it takes to get a global guideline implemented. The path to the guideline harmonization and delivery spans over 6 years of intense and successful collaboration between 9 regulatory agencies and 6 industry organizations. The expert working group's journey went through a 5-step harmonization process to reach the summit, the current stage of the implementation of this guideline. Let's now take a look at each step. As you see in this slide, at base camp, step 1, the topic was endorsed by the ICH Assembly in June 2018. Then, until November 2019, the expert working group worked on several technical documents, of which a proposal for a new guideline. Several topic subgroups were also formed at this time. The path to Camp 2A was reached in November 2021. During 2 years, expert working group members and subgroups worked on the development of the draft guideline. Multiple monthly ad hoc teleconferences, in-person alignment meetings, and internal consultations were held to build consensus around different topics. In June 2022, the expert working group celebrated the release of the ICH M12 draft guideline after its adoption by regulators as Step 2B. This opened the start of the public consultation phase. In November 2022, the public consultation call for input to the draft guideline was complete. 1 year later, in November 2023, the EVG and its 10 topic-based subgroups compiled and addressed over 1,000 comments received from drug industry and other subject matter experts. In terms of harmonization, the group built upon areas of existing regional DDI consensus, shared insights, and identified potential solutions to topics for which there was no initial consensus. In March 2024, the ICH M12 harmonized guideline was signed off by regulatory topic leaders and adopted by the ICH Assembly as Step 4. The summit, Step 5, was reached in May 2024 with the release of the ICH M12 guideline and the beginning of the implementation phase. This led to the adoption of the guideline by the FDA in August 2024 and the hosting of several training forums. Now that we have a new FDA guidance adopted as of August 2024, what is the implication for FDA's 2020 final guidances for you? This new guidance replaces the following 2 guidance that were published in January 2020. In vitro drug interaction studies and clinical drug interaction studies both focused on cytochrome P450 enzyme and transporter mediated drug interactions. The current final FDA guidance is accessible at the following link on the FDA website. I would like to thank you for your interest in DDIs and harmonized recommendations for their application. I will now hand it over to Dr. Young, who will share insights on in vitro DDI assessments. I will be back at the end for the Q&A. Thank you.
Thank you, Helen, for the great introduction. So herein, I will talk about in vitro DDI assessments, followed by a short Q&A before we move to the presentation by Dr. Kellie Reynolds talking about clinical DDI assessment. Then we will have another Q&A session. So here is the outline. In this presentation, I will talk about investigational drug as a substrate, inhibitor, or inducer of a CYP enzyme or drug transporters. I will also talk about DDI liability of metabolites and UGT-mediated DDI. So first of all, I would like to thank all the members of the M12 expert working group, including several of my colleagues from Office of Clinical Pharmacology. Dr. Matabushi is the rapporteur of the group. Dr. Reynolds is the topic lead. and Helen is a rapporteur supporter, with another of my previous colleagues serve as the previous rapporteur supporter. Also, I would like to thank several of my colleagues who are the subject matter of experts in our office. And I would like to thank the support from our management, Dr. Zane, who is the Director of Office of Clinical Pharmacology, and Dr. Xiumei Huang, who is the Deputy Director. So here I listed some topics that have updated in the M12 guideline compared to FDA DDI guidance. The endogenous biomarker one will be described by Dr. Reynolds in her presentation. And in my presentation, I will briefly talk about plasma protein binding and some change in the simple criteria to evaluate in vivo DDI potential of a drug from in vitro data, and also the DDI liability of metabolites as an inhibitor or inducer, and the UGT-mediated DDI. And in addition, the appendices of M12 guideline has a list giving example of drug used for in vitro or in vivo studies as a substrates, inhibitors, or inducers of CYP enzyme, UGT enzyme, or drug transporters. Please note that these are just examples and do not mean to be a comprehensive list. So a company can use the other drugs with justification based on their experience. First, let's talk about the timing of in vitro DDI evaluations. This is usually expected to be conducted early during drug development. The results will determine the need and the timing of clinical DDI studies, and to inform the decision on exclusion criteria or prohibited medication of clinical trial protocols. On one hand, we want to make sure the safe use of drugs. On the other hand, we want to avoid unnecessary exclusion of medication. In vitro data generally recommended to be available before starting study in patients who likely take multiple drugs together and have the potential for drug-drug interaction. The DDI potential of metabolites may be evaluated later in development when more knowledge about metabolites become available, for example, from a mass balance study in humans. So let's look at the in vitro evaluation of a drug as a substrate of enzymes. This is usually conducted by metabolic phenotyping experiment to look at several major CYP enzymes. If the above CYP enzymes do not play a major role, then we will consider the involvement of other enzymes, such as some other CYP enzyme or other phase 1 enzymes, and sometimes phase 2 enzymes if there is evidence suggesting direct glucuronidation or sulfation of a drug. If an enzyme is found to be responsible for 25% or more of the elimination of a drug, then we may consider further clinical evaluation, that is, evaluate the effect of an inhibitor and or inducer of that enzyme on the PK of the investigational drug. On the other hand, we also want to know if an investigational drug can affect the metabolic enzyme. For the CYP inhibitor potential, these are the major CYP enzyme we usually look at. For CYP inducer potential, we usually start looking at CYP1A2, 2B6, and CYP3A. So after the results from this in vitro experiment become available, we will use some simple model as described later on to make the initial assessment. If we cannot rule out the DDI potential in vivo, then companies have the choice either go directly to conduct clinical DDI studies, or they may use a more advanced approach, modeling approach, either statistical modeling and or dynamic modeling, often called as a PPK, to make a more quantitative prediction, and then decide whether there's a need to conduct a clinical DDI study. Before talking about the simple method, I would like to briefly talk about plasma protein binding determination. Unbound drug concentration are usually used in the in vitro to in vivo extrapolation criteria. In the past, the guidance is recommended capping the free fraction of drug in plasma FUP at 0.01, even if a compound has plasma protein binding ppb more than 99%. This comes from a concern about the uncertainty in the measurement of ppb for very highly protein-bound drug. While such a practice can reduce false negative, it also leads to some false positive prediction that can translate into unnecessary clinical DDI studies or unnecessary restraints on the use of concomitant medication in patient studies. This has long been discussed about the need to cap FUP at 0.01. Based on the review of recent literature and the presentation from experts, the working group decided that with the advances in methodology, including bioanalytical method, the ppb for very highly protein-bound drug could be reliably measured. So that in the M12 guideline, flexibility is provided, allowing companies to use experimentally determined ppb. That is, FUP can be below 0.01 in the IVIVE criteria if the accuracy and the precision of ppb measurement has been demonstrated. However, if the reliability of FUP measurement less than 0.01 cannot be demonstrated, a default value of 0.01 should still be used. So first, let's look at how to determine whether a drug inhibits CYP enzyme in vivos using the basic model. For reversible inhibitors, basically we compare the unbound Cmax of a drug at a therapeutic dose divided by unbound Ki, compare the ratio to a cutoff value of 0.02. If the ratio is less than this cutoff value, then we can rule out the in vivo DDI potential. The Ki can be experimentally determined or calculated based on IC50 and the substrate concentration using Chen-Prosoff equation, or sometimes roughly estimated by using half of the IC50 value. Ki unbound is unbound inhibitor constant, which is Ki multiplied with the free fraction of drug in microsome or hepatocyte preparations. This free fraction can be experimentally measured or sometimes estimated in silico. For CYP3A inhibitors, because of the significant presence of CYP3A in the intestine, we also need to consider the potential of intestinal CYP3A inhibition, so that the in vivo potential can be excluded if the dose divided by 250 milliliter divided by the Ki compared to a cutoff value of 10. This dose divided by 250 milliliter is a rough estimate on the intestinal inhibitor concentration. And this could be an overestimation for drugs with low solubility, such as BCI class 2 or 4 drugs. Some inhibitors are also time-dependent inhibitor of CYP enzyme. The basic model is listed here. The K big I and the K inact are inhibitor potency parameters of a compound determined experimentally. The K degradation is the turnover rate of affected enzyme. The appendices of the guideline provide a commonly used value for individual CYP enzyme. And for CYP3A, there are 2 Kdec values in the liver and in the intestine, respectively. Please note that the scaling factor applied to the unbound Cmax is reduced from 50 times in the FDA guidance to 5 times in the current M12 guideline. This change was based on recent literature studies and the reanalysis of data published previously, both of which suggest no increase in the false negative predictions, while this can reduce false positive prediction when we reduce the scaling factor. So now let's talk about how to determine whether a drug can induce CYP enzyme in humans. As mentioned earlier, this usually starts from evaluate the induction potential of a drug on CYP1A2, 2B6, and 3A initially. If no induction of CYP3A is observed, then there is no need to further evaluate the induction potential of a drug on CYP2C enzyme, because CYP2C enzyme are considered also regulated by the drugs, the inducer for CYP3A, and they are generally less inducible compared to CYP3A. If a drug induces CYP3A, then there may be a need to further evaluate the drug potential to induce CYP2C enzyme. The guideline also mentioned the phenomena of downregulation, but a clear recommendation cannot be provided due to very limited. To evaluate the induction potential of a drug on CYP enzyme, we usually use mRNA data because activity measurement can be confounded by inhibition if a drug is also an inhibitor. In addition, mRNA of CYP3A usually has large changes than activity, so that mRNA data are more suitable for categorizing Emax and EC50. There are several methods to evaluate the mRNA data. For the sake of time, I will focus on the mRNA photo of change method, which is most often used. So in vivo induction potential of a drug cannot be readily excluded if the drug in hepatocyte from at least one donor meets both of the following criteria. One is a concentration-dependent increase in mRNA expression of a CYP enzyme. The other is the fold change of the CYP mRNA expression relative to the vehicle control is twofold or more at a concentration approximately 50 times of unbound Cmax of a drug, or at lower concentrations. So please note that the scaling factor in the FDA guidance is 30 times and 15 times in the draft version of M12 guideline. And in those guidance, the FUP was capped at 0.01. This scaling factor is changed to 50 times in the final M12 guideline based on the data from a literature study published by a pharmaceutical consortium innovation and quality IQ induction working group, and also internal analysis of data from NDAs approved by EMA and the FDA in recent years. The increase of the scaling factor is intended to mitigate false negative predictions when using FUP below 0.01 for certain inducer drugs that have a plasma protein binding more than 99%. The response to positive control is usually expected to be sixfold or higher. When the response to a positive control is less than sixfold, induction potential of an investigational drug cannot be easily ruled out, even if it increases the CYP mRNA less than twofold of vehicle control, but more than 20% relative to the positive control. In such situations, further evaluation is needed. And the guideline include an equation on how to calculate the response of the positive control for a tested drug. It is not uncommon that even positive control have limited induction effect on CYP2C enzyme. This makes it challenging to interpret the in vitro data for tested drug. An approach is to leverage in vivo DDI study results with a sensitive CYP3A substrate, since CYP2C enzyme share the regulatory pathway with CYP3A but are less inducible. This approach will be described more by a later presentation by Dr. Kellie Reynolds. In addition, I want to mention that there's no clear recommendation on how to evaluate enzyme activity data in the induction experiment. This will need more research and evaluation. But for CYP2C19 enzyme, somehow its mRNA is not sensitive to induce even positive control. So it's hard to rule out induction potential of a drug. In the M12 guideline, it recommends measurement of CYP2C19 activity. So now let's switch the gear to about the transporters. For drugs as a substrate of transporters, in vitro experiments are usually conducted for several transporters. One set is a PGP or BCRP, both are efflux transporter. These are often conducted for orally administered drug because PGP or BCRP can limit drug absorption. This will be also considered when biliary excretion or renal active secretion is likely a major elimination pathway of a drug because PGP or BCRP can mediate biliary excretion or renal active secretion. And when the pharmacology target of a drug is in the brain, such an experiment is also informative because PGP or BCRP present on the blood-brain barrier can limit the brain tissue penetration. OATP1B1, 1B3 are the major hepatic uptake transporter. So in vitro experiment to evaluate the drug as a substrate of this transporter is needed if hepatic metabolism or biliary excretion accounts for at least 25% of the elimination of a drug, or the pharmacology target of a drug is inside the liver. There are several renal transporters, OAT1, OAT3, OCT2, which are uptake transporter, and MATE1, MATE2K, which are efflux transporters. In vitro experiment need to be considered when the drug has a significant active renal secretion, defined as accounting for 25% or more of systemic clearance of a drug. The guidelines include the equation estimate the renal active secretion clearance based on assumption that the passive reabsorption is equal to passive secretion, and thus there's no active reabsorption. Additional transporter may be considered case by case, for example, efflux transporter MRP2, or intestinal uptake transporter OATP2B1, and hepatic uptake transporter OCT1. For drug as inhibitor of transporters, here are the major transporters usually to be evaluated. And based on the in vitro study results, such as IC50, we will apply some simple equation to make an initial assessment. Similarly to CYP enzyme inhibition, if we cannot rule out the potential of in vivo interaction, the companies can either conduct a clinical DDI study or use a modeling approach to make further evaluation. And the table here listed the criteria to evaluate the in vivo inhibition potential of a drug on the major drug transporters. The criteria are basically the same as those in the FDA guidance, except that the cutoff value for mid transporter is reduced from 0.1 to 0.02. And for a drug administered non-orally, or if it is the metabolite of a drug that is formed post-absorption that inhibit PGP or BCRP, we will consider evaluate the inhibition potential by looking at the unbound Cmax divided by IC50 and compared to a cutoff value of 0.02. The other cutoff value can also be used if these are justified based on IV-IVE analysis and a calibration of the in vitro system with known inhibitors and non-inhibitors of transporter systems. So for the drugs as an inducer of transporter, in vitro methods to evaluate this are not well established at this time. Therefore, recommendations for the in vitro evaluation of a drug as a transporter inducer are not provided in the guideline. An approach is to leverage in vivo DDI results with a sensitive CYP3A substrate because PGP share the same regulatory pathway with CYP3A, but is less inducible. Please see the next presentation by Dr. Reynolds for more. So now let's talk about the DDI liability of. So first of all, in some situations, we need to evaluate the metabolic phenotyping of metabolite, that is, how metabolite is further eliminated. So this will apply to metabolites that significantly contribute to the overall efficacy, meaning that the metabolite contributed to efficacy more than or similarly as a parent drug. The contribution is estimated based on the exposure of a metabolite relative to the parent drug based on molar units, and also consider the free drug concentration. Also, it will consider the pharmacology potency, for example, receptor binding affinity or enzyme inhibitory potency. and also the data related to targeted tissue distribution of a metabolite and the parent drug if such data are available. On the other hand, if a metabolite are suspected to cause a significant adverse event based on non-clinical or clinical information, there may also be a need to evaluate how the metabolites are further eliminated. For example, sometimes certain metabolites contribute to QT prolongation. The above consideration generally also apply to metabolites as transporter substrates. Metabolites may also carry DDI liability as an inhibitor of CYP enzyme and transporters. There are some update in the ICH M12 guideline compared to the FDA guidance. In the previous FDA guidance, it recommended considering both the exposure of a metabolite relative to the parent drug and also compare the polarity of a metabolite to parent drug. But the ICH M12 guideline adopted a pragmatic rule. It recommended to evaluate metabolites inhibition on CYP or transporters if the metabolite meet both criteria. One is it determined as a major metabolite per the MIST guidance, which means that it accounts for at least 10% of the drug-related material in circulation, which is often judged based on the results from a mass balance study, and also the metabolite has AUC of 25% or more relative to the parent drug. This change was made based on analysis in a recent publication, which looked at the polarity of a metabolite and its inhibition potency compared to parent drug for a group of hydroxy metabolite or methyl or diethyl metabolite. The analysis did not reveal a clear relationship between the polarity and inhibition potency. So the M12 guideline uses this approach, which is actually adopted from the recommendation in EMA guideline. This will help reduce the excessive work for situation where a drug is extensively metabolized and it will have a number of metabolites which can easily exceed the exposure of 25% or even the exposure of the parent drug. Based on the results of an in vitro study of a metabolite, whether to conduct a clinical DDI study will be determined with the same approach as those for parent drug. Metabolites may not be relevant for intestinal CYP or transporter inhibition unless metabolites are formed substantially in the gut or intestinal cells. And when a parent drug is found to inhibit a CYP enzyme in vitro and predicted to have such potential in humans, a clinical study may be decided to be conducted. In such case, there is no need to perform separate in vitro experiments to evaluate the inhibition potential of a metabolite on the CYP enzyme affected by the parent drug, because the clinical study will cover the effects of both parent and metabolite. But there are some exceptions where when the clinical relevant exposures of the metabolite cannot be adequately reached in the clinical DDI study. For example, the study duration does not allow the metabolite to accumulate when the metabolite has a longer half-life than the parent drug. However, when a model, either mechanostatic or PPK model, is used to predict the inhibitory effect of a drug on CYP enzyme to replace a clinical DDI study, the contribution of a metabolite need to be incorporated in the model. In addition, metabolite may also act as an inducer of a CYP enzyme. Herein, we assume that generally the effect of a parent drug in the in vitro induction experiment may cover the effect of metabolites, since metabolite may be generated during the incubation of a parent drug with hepatocyte usually lasts for 2 to 3 days. But there are some exceptions. if a drug is a prodrug or a metabolite, is mainly formed extrahepatically, separate in vitro evaluation of a metabolite induction potential on CYP enzyme is recommended for metabolites that are considered as a major metabolite and also has AUC relative to patent drug for 25% or more. So lastly, I want to spend a few more minutes talking about UGT-mediated. So first, when a drug is mainly eliminated by direct glucuronidation in humans, we recommend in vitro experiment to categorize which UGT isoform are responsible for the glucuronidation of the drug. On the other hand, there's also possibility of investor drug as a UGT inhibitor. While a drug can be an inhibitor of an enzyme, even if it is not a substrate of the enzyme, we consider routine evaluation of any drug to inhibit UGT may not be needed. This is considering the overall limited magnitude of drug interaction mediated by UGT inhibition, because based on the literature of clinical DDI study, the AUC increase of UGT substrate generally not exceed threefold. A lot of time is twofold or less. And also, we consider the challenge with predicting in vivo DDI potential from in vitro data for UGT inhibitor. So the guideline recommend in vitro studies for a drug whose major inhibition pathway is direct glucuronidation to look at its inhibition potential towards some UGT isoform. The IVIV criteria has not been established for UGT like the one has been set up for CYP enzyme or transporter. This is because limited available data for UGT inhibition. So further research is warranted to explore such a IVIV criteria. And the companies may consider using the same criteria as the one for CYP reversible inhibitors, or they can propose an additive with justification. In addition, when an investor drug will be commonly administered with a drug that is mainly metabolized by direct glucuronidation, we recommend in vitro experiment to evaluate the inhibition effect of investor drug on the UGT isoform mainly responsible for the elimination of the other drug. Lastly, I want to mention that in vitro experiment and bioanalytical methods are not necessarily GLP standard, but bioanalytical assays should meet some general requirements to ensure reliable measurements. And in addition, we need to pay attention to several factors that can cause a significant difference between actual drug concentration in the in vitro assay to nominal concentration, such as poor aqueous solubility of a drug, cause a precipitation or instability of a compound in the in vitro assays, or non-specific binding of the drug, for example, with the well of the plate or tubes. Correction for binding or stability or solubility issue need to be considered when interpreting the in vitro data. And finally, we have made a tremendous advancement to enhance our understanding of drug-drug interaction mechanism. Collective effort from industry, academia, and regulatory agencies led to a globally harmonized DDI guideline. While we have reached consensus on a number of important topics, there are still areas that warrant further research, which will improve the efficiency of DDI assessment during drug development, including several topics I mentioned earlier. For example, how to further reduce false positive prediction for time-dependent inhibitor for inducer of CYP enzyme, and also for CYP3A reversible inhibitor or PGP and the BCRP inhibitor, which has low solubility, so that the dose divided by 250 milliliter may overestimate their intestinal inhibition concentration. And also, we still have much fewer data for metabolite DDI compared to parent drug. And also, we need to explore the in vitro to in vivo criteria for UGT enzyme. So finally, I want to thank for your attention, and we can answer some questions. Thank you.
All right. Thank you to our presenters for their wonderful presentations. We will now begin our first question and answer session. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. So we have some questions that have already come in. We'll do a first question here for Helen Heyman. Helen, was PMDA Japan one of the 9 regulatory agencies?
Yeah, thank you so much for this great question. Yes, the Pharmaceuticals and Medical Devices Agency in Japan is one of the 9 regulatory agencies that has been involved in the ICH guideline. It might perhaps be of interest that the chair of the expert working group is from PMDA. Thank you.
Thank you so much for that response. We have another question that came in for you as well. Has the ICH M12 been fully adapted by FDA at the moment?
Oh, yeah, this is an excellent question. So, yes to this one, the ICH M12 has been fully adopted by the FDA in August 2024. And with that, it is replacing the 2 previous guidances of January 2020, if you recall from the presentation, namely the in vitro drug interaction studies and the clinical drug interaction studies. Both focus on cytochrome P450 enzyme and transporter mediated drug interactions. Thank you.
Okay, thank you as well for that clarification. So now we'll address a question for Dr. Shining Zhang. Dr. Yang, you advised that in vitro DDI studies should be conducted early in development before going into patients. Would this usually be after FIH studies?
Yeah, that's a good question. I would like to clarify that quite a lot of in vitro DDI study prefer to be conducted before first in human studies. For example, many drugs are eliminated by metabolism, so we would like to get an early idea about how the drug is eliminated by CYP enzyme. So the metabolic phenotyping study are usually conducted early, and also this is useful to help predict the PK of drug, which will help to decide whether a drug will be bring into the clinical studies, including first in human studies.
All right, thank you for that clarification. We'll have another question for you, Dr. Yang. For CYP induction, the 50x CU max hits everything. Is there any plan to revise it to lower, like 30x?
Yeah, that's an excellent question. As some of you may notice that this scaling factor, has changed among the regulatory guidance. 50 is in EMA guideline, 30 is in 30 in FDA guidance, and 15 in ICH chapter guideline, and now we end up with 50. I want to say that it hit everything. Still it can rule out a number of drugs. I think this is an area need further research. Apparently, induction is more complicated than inhibition. One possible way is to consider the response to the tested drug relative to the positive control. For example, rifampin for CYP3A. If the induction of mRNA, it's just exceed twofold, let's say threefold, but it's much less than that of positive control. That kind of implying that this hepatocyte is very sensitive to induction effect. This may not necessarily translated into in vivo. However, this needs some data analysis, to demonstrate what's the kind of a threshold of the response types to the drug relative to the positive control. I saw that in the question. There's another thought is that could we apply the different scaling factor, to drugs with very highly protein bound versus not that highly protein bound. I think that's potentially could be a possible solution. Again, this needs data analysis across a relatively large number of compounds to show that this can maintain this can still control the false negative while reduce false positive.
All right. Thank you so much for that informative response. We will now address a question that came in for Helen Heyman. Does the new ICH M12 guidance fully supersede the 2020 FDA DDI guidances? Should drug development programs henceforth only reference the new ICH guidance, for example, in submission documents?
Yes, this is an important question. Thank you. So yes, the new ICH guidance does fully supersede the 2020 FDA guidances I was referencing previously. So it does fully replace them in terms of, for example, so for submission documents, yes, you would be referencing the new guidance. Thank you.
All right, thank you for that clarification as well. Now we'll go to another question for Dr. Yang. What is your recommendation on how to use the data for the measured drug concentration in vitro induction assays? Sorry, Dr. Yang, if you're speaking, you might be on mute.
Okay, sorry.
Oh, there you are.
Yeah, thank you. This is another great question. We got comments on this in the past. I think the intention is mainly concerned with large difference between the nominal concentration and the drug concentration in the experiment. Because if there's a large difference from the actual concentration, nominal concentration, often we use a nominal concentration, right? So this could lead to under predicted DDI potential. For this, first of all, we may think about some possible reasons for that. One is instability of a compound, or solubility, limited solubility cause precipitate drug, or non-specific binding of drug to the apparatus, such as the well of the plate. So this, should have been examined early in the pilot experiment for the test compound. If such thing happened, it reached the flag, then we either need to adjust the experiment condition, reduce this confounding factor, or take this into consideration to correct the in vitro results we interpreted the data. As to measure drug concentration in the induction assay. Well, this need to think about if we observe the difference, we also need to think about what may be the reason. If this is caused by metabolism or transport of the drug, then this may just mimic the in vivo situation. So this is an area still need to further investigation into this. In the past, people do not pay a lot of attention to this, but I would say some of the things I mentioned earlier, instability, precipitation because solubility issue, and non-specific binding, those should be pay attention to and easily be examined in pilot experiment.
All right, thank you. Another question for you, Dr. Yang. Are all these DDI testings you have presented required before phase 1, or can some of them be done at a later stage?
Yeah, this is a great question. It's kind of related to an early question, but I will say no, it's not always required before phase 1. Let's use transporter study as an example. As I mentioned, for drug as a substrate of renal transporters, usually we expect the significant interaction happen just for those drugs have a significant renal active secretion. Apparently, before the phase 1 study, we won't really know how much renal elimination play a role in the elimination of the drug. So usually from the early PK study, we collect the urine sample, so we will have a rough idea about whether there's a significant renal elimination. Then we can make a rough estimation on whether there's a substantial renal active secretion. So in such case, you can see that the in vitro experiment testing a drug as substrate of renal transporter doesn't necessarily to be done before phase 1.
Thank you so much for that response. We can do another question for you, Dr. Yang. Can substrate assessment for OATP1B1 3 be skipped if a compound is cationic?
Yeah, that's a great and interesting question. So as the audience reads the question, must notice that in this guidance, we removed that compared to some earlier version of FDA guidance. It used to think that OATP1B, from the name, organic anion transport peptide, transport anion compound. That's often the case, but still we came across some drugs that are not anionic compound, but still substrate of OATP1B transporters. So if hepatic elimination play a major role, or the pharmacology target of the drug in the liver, I think it's still prefer that this is being examined whether a drug is substrate of OATP1B.
All right, thank you for clarifying that. Now let's go ahead and address a question that came in for Helen Heyman again. What is the M12 in the title of the document?
Oh, that's a very good question. M stands for multidisciplinary. There are different categories of documents in ICH. And what the 12 means is since the inception of ICH in 1990, the M12, the DDI document, is the 12th in the category of the multidisciplinary documents. Thank you.
All right, thank you so much for clarifying that. We'll now go to a question that came in for Dr. Yang. If the calculated value exceeds the cutoff value, can the obtained value used to rank the drug as weak, moderate, or strong inhibitor? Or if so, which value ranges?
Yeah, that's a great question. Very interesting question. I don't think there's such a cutoff value. The thing is that this simple criteria compared to cutoff value is more used for qualitative assessment. If it's below the cutoff value, we can readily rule out the DDI potential. So it's definitely worthwhile to examine whether there's also a quantitative correlation. I mean, I expect that in general there will be such a trend for the ratio. If it's high, then there are more likely it lead to a stronger interaction. But I don't know how well such a correlation is, so that it can warrant to tell whether this will be a weak, moderate, or strong inhibitor in vivo.
All right, thank you for that information. We have another question for you, Dr. Yang. For CYP3A induction potential, if the developmental compound has a short half-life and is intended to be washed out during each dosing interval, in vitro induction, say with 72 hours of continuous exposure for hepatocytes, may it overestimate induction in vivo? So I guess the question there is the absence of effect on CYP3A biomarkers and absence of autoinduction sufficient to prove absence of in vivo induction?
Yeah, this is also a great question. yeah, actually, there are quite a lot of false positive prediction from in vitro data using the different methods to interpret mRNA data. So this question raised interesting one about the CYP3A biomarker. Actually, in the this ICH guidance, we do mention about the utility of biomarkers. CYP3A biomarker, I think among those 4-beta hydroxycholesterol is the most studied ones. As suggested by several published papers, I think there's some utility of this biomarker. I remember that the publication showed that this biomarker could be used to tell whether an in vitro inducer can be a potent inducer in vivo, means that it will be a strong inducer or a moderate inducer. Although it's not as sensitive as midazolam, so that this may not rule out the possibility of the drug as a weak inducer of CYP3A. But I think this can, at least help to tease out the possibility of the drug be a significant potent inducer early.
All right, thank you so much. We might have time for just one more question in this session for you, Dr. Yang. How soon in vitro liabilities of transporters are they to be checked?
Yeah, this kind of come in line with early question. This because transporter is more complicated than CPA enzyme, which is mainly in the liver. Our CYP3A also has significant presence in intestine, but the transporter they present in different tissues. So some of this need to consider the administration route of the drug, the elimination pathway, and the target site. So yeah, it's need to based on totality of factors, then determine whether a transporter may play a significant role. So that then some of this information sometimes need to come from a clinical study. So the timing for transporter study, I think, vary more than CYP studies.
All right, thank you again for that response. So that looks like that's all the questions we have time for this first Q&A session, but please note our presenters here will return for the following panel that will happen after the next presentation. Oh, sorry. So I'll go ahead and introduce our next presenter here. So now we will be hearing from Dr. Kellie Reynolds, Director of the Division of Infectious Disease Pharmacology within the Office of Clinical Pharmacology, that's within the Office of Translational Sciences. So please join me in welcoming Dr. Reynolds.
Hello, and thank you for your interest in evaluation of drug interactions during drug development. I will provide an overview of the clinical drug interaction information included in the ICH M12 drug interaction final guidance. The outline lists the topics I will discuss. Due to the level of detail in the guidance, I will not address all clinical topics. I will start with the foundational topics of study types and spend the majority of my time on topics that either represent a change from prior practice or topics that generated a lot of comments during public consultation. During the question and answer period, you can ask questions about any topics in the guidance. I will start by framing the scientific evaluation of DDIs in the context of a drug development program. As stated in the background section, this guidance provides general recommendations on how to evaluate the DDI potential of an investigational drug. It is recognized that the DDI evaluation is generally tailored based on the specific drug, intended patient population, and therapeutic context. Alternative approaches may be acceptable if properly justified. Thus, the scientific rigor described in the guidance needs to be considered with the patient population in mind. The end of the process is useful information for the healthcare provider and patient. For some drug development programs, evaluation of DDIs is straightforward. do the basic in vitro studies, which lead to the conduct of a few clinical studies that provide a clear picture of DDI liability and instructions for patients. Other programs are more complicated. It may be difficult to determine which clinical studies are most informative. The guidance instructs drug developers on how to go through the drug interaction program and consider the most appropriate next step. There are several possible goals to consider for a given DDI study. The overarching goals are to determine the presence or absence of a DDI. Does drug A interact with drug B? The magnitude of the DDI, if one exists, and considering the magnitude, is a DDI management strategy needed? However, there is a more nuanced purpose that led the study to be conducted in the first place. Some studies are conducted to define the DDI liability by a specific pathway. For example, do strong CYP3A inhibitors cause a clinically significant increase in drug exposure? Other studies refine the understanding of DDIs in clinical use, and some studies may serve both purposes. This table lists the various types of clinical studies discussed in the guidance. I will discuss all of them except for cocktail studies as I go through my presentation. Standalone studies are the typical Phase 1 DDI study. Nested studies evaluate DDIs as a subobjective of a larger study. Studies with index precipitants or substrates typically define the greatest magnitude of interaction by a specific pathway. Studies with expected concomitant drugs investigate interactions between a drug and other drugs likely to be used by patients. Cocktail studies investigate the effect of the drug as a possible inhibitor or inducer of multiple enzymes or transporters. The final guidance describes an approach that uses endogenous biomarkers that are substrates for drug metabolism or transport. This approach was not addressed in detail in the regional guidances and guidelines. The guidance provides general considerations for selection of substrates, inhibitors, and inducers for use in clinical DDI studies. When selecting the drugs, consider the goal of the study. Studies with index precipitants and index substrates estimate the greatest magnitude of interaction, and the results can be extrapolated to other drug combinations. Studies with expected concomitant drugs are often based on mechanistic understanding of DDI potential, which inform the selection of specific drugs. The studies provide useful clinical information for a drug pair. However, it may be difficult to extrapolate to other drugs. With careful selection and an understanding of possible DDI mechanisms, some extrapolation is often feasible. And note that index drugs are not available for transporters and UGT enzymes. The process for evaluating CYP-mediated interactions is similar to the process described in regional guidelines. When evaluating a drug as a CYP substrate, it is typical to start with a strong index inhibitor and strong index inducer. However, the guidance mentions there are some scenarios where starting with a moderate inhibitor or inducer may be informative. For example, if it is expected that a strong inducer will significantly decrease drug concentrations to a degree that precludes co-administration in patients under any circumstances, it may be more valuable to start with a moderate inducer. When evaluating a drug metabolized by a polymorphic enzyme, PM versus EM evaluation may be informative. When evaluating a drug as a CYP inhibitor or inducer, start with a sensitive index substrate, but consider the selectivity of index drug for the enzyme to aid the selection of substrate and interpretation of results. The M12 guidance provides general considerations on the assessment of UGT-mediated interactions, going beyond the passing mention in the regional guidelines. Clinical studies of a drug as a substrate of UGTs are recommended on a case-by-case basis, considering the safety profile of the drug and the likelihood of its concomitant use with inhibitors. Similarly, it is recommended to evaluate a drug as an inhibitor of UGTs on a case-by-case basis, considering likelihood of the drug's administration with substrates and the safety profile of those substrates. Considerations for studying a drug as an inducer of UGTs is more complex. There is limited understanding of gene expression of UGTs. UGTs are co-regulated with CYP3A but are less inducible. After much internal discussion and consultation with experts, the working group recommended the following. If a drug reduces AUC of a sensitive CYP3A substrate by more than 50%, the need for a clinical DDI study with UGT substrates is determined based on the following considerations. The magnitude of the CYP3A substrate decrease when co-administered, the likelihood of use with UGT substrates, and whether the UGT substrates are also substrates for other enzymes or transporters regulated by PXR or CAR. During the public consultation period, commenters stated that the 50% reduction in CYP3A substrate criterion would lead to unnecessary clinical studies. However, the guidance indicates that the 50% reduction begins the case-by-case consideration, which may not always lead to a clinical study. I will move on to the discussion of considerations for clinical studies of transporter-mediated interactions. The guidance includes this table that describes when to consider clinical evaluation of a drug as a substrate of transporters, depending on the absorption, distribution, and elimination characteristics of the drug. If a clinical study of the drug as a transporter substrate is needed, the selected inhibitor drug for the study should be a known inhibitor of the transporter under study. Because there are no index inhibitors for transporters, the inhibitor is generally selected based on likely concomitant use. The M12 appendix includes examples of transporter inhibitors, but it is not a comprehensive list, and it is acceptable to use other inhibitors. When selecting the inhibitor, it is important to consider other enzymes or transporters involved with the drug's ADME to ensure study results will be interpretable. The need to conduct a clinical study of a drug as a transporter inhibitor is based on likely concomitant drugs that are substrates for the transporter and the safety considerations for those drugs. Preferred substrate drugs are those whose pharmacokinetic profile is markedly altered by co-administration of known inhibitors of the transporter, and it is useful to select a likely concomitant drug if possible. Endogenous biomarker studies can be informative to address this drug interaction mechanism, and I will provide more information later in the presentation. The transporter induction section of M12 only mentions PGP based on limited data for induction of other transporters. PGP is also co-regulated with CYP3A and is less inducible than CYP3A. Thus, the considerations for evaluation of a drug as a PGP inducer are almost identical to the considerations for evaluation of UGT induction. As I mentioned at the beginning of this talk, the M12 guidance includes a section on use of endogenous biomarkers in drug interaction assessment. Details were added based on encouragement during the public consultation period. The working group consulted with experts and reviewed relevant literature when writing this section. These studies provide an alternative approach to assess a drug's potential as a precipitant, evaluating the change in exposure of a well-characterized endogenous substrate. The assay for the analytical method needs to be validated. At this time, few endogenous biomarkers are validated and characterized in terms of their performance characteristics. The guidance provides specific details describing the use of CP1 for evaluation of hepatic OATP1B inhibition potential. The recommendations are supported by recent literature reports. The assessments can be incorporated into early healthy volunteer studies. To use the method, plasma CP1 is measured prior to drug administration, providing a baseline. Baseline AUCT is calculated as baseline CP1 times T. Next, serial samples post-drug administration allow characterization of Cmax and AUC. The post-drug administration to baseline ratio for AUC and Cmax are calculated. A ratio less than 1.25 indicates a low likelihood of a clinical DDI by OATP1B inhibition. My next topic is nested DDI studies, which is the evaluation of DDIs as part of a larger study, usually a phase 2 or phase 3 study. The advantages of nested DDI studies include the fact that they are conducted in the clinically relevant population and may represent the anticipated clinical setting. Of course, there are challenges related to study design and data collection. Additionally, these studies typically evaluate the investigational drug as an object, but not as a precipitant. It is important to prospectively design nested DDI studies to maximize the chance of generating useful results. Investigators should prospectively plan all aspects of the DDI assessments. This includes pre-specifying the concomitant drugs or groups of drugs to evaluate, using simulations to assist selection of sampling times and number of samples needed, and ensuring accurate data collection, the time of drug administration, sampling, food intake, and other concomitant drugs. And it should also include developing a population PK analysis plan. While unplanned retrospective analyses are not ideal, the working group recognized they may occasionally be conducted because of observed safety or efficacy issues or new DDI concerns. The degree to which an observation can be linked to a pharmacokinetic interaction depends on the availability of plasma drug concentrations and other information typically included in a prospective plan. In most cases, DDIs identified or ruled out through a retrospective analysis need to be confirmed using a prospective evaluation. The next group of topics are related to interpretation of study results and determining the appropriate next actions. When interpreting a study designed to evaluate the investigational drug as the object of a DDI, the point estimate of the ratio between the exposure of the object with and without the precipitant can be used to describe the magnitude of the interaction, and this has been the standard practice. The preferred approach to interpreting the point estimate is to use no-effect boundaries that are developed based on exposure-response relationships from clinical use. The variability of drug exposure in the indicated population is incorporated into the consideration. The working group discussed the utility and need for the 80 to 125% acceptance range, similar to that used in bioequivalence studies. While the guidance mentions this approach to no-effect boundaries, it is typically overly conservative and not necessary or recommended. When interpreting studies that evaluate the drug as a potential precipitant of DDIs, there is no change from the classification system for CYPs. Comments we received during the public consultation period requested a similar system for transporters. The working group anticipated this interest and considered the feasibility when writing the draft. However, because the interaction mechanisms for most substrates and inhibitors involve other transporters or enzymes, the establishment and use of a classification system remains challenging. It is not possible to study all combinations of drugs, so extrapolating results from a drug interaction study to another group of drugs is an essential component of a drug interaction program that benefits patients. There are many complex scenarios, interactions that involve multiple pathways, or those that combine with organ impairment. Knowledge of ADME properties for the investigational drug and potential concomitant drugs assists the extrapolation, and mechanistic modeling can also assist extrapolation. The predictive modeling appendix to ICH M12 indicates how mechanistic modeling approaches can be used to characterize the potential for DDIs. Also, they can indicate whether a dedicated clinical DDI study is needed and support clinical recommendations in the absence of a clinical DDI study. Some of these uses are related to extrapolation, which I mentioned on the previous slide. Multiple approaches for assessing DDI risk may be feasible. There is not a required progression of in vitro evaluation to mechanistic modeling to a clinical study. As results emerge and are interpreted, a variety of additional study types can inform next steps. Importantly, any model used for quantitative prediction requires use of appropriate in vitro experimental conditions. The guidance provides information about the use of mechanistic static and PBPK modeling because both approaches are useful. Mechanistic static models incorporate detailed drug disposition and DDI mechanisms for precipitant and object. They may estimate the effect of several combined interaction processes. For interpretable results, the input parameters should be justified by data or literature. The most frequent use is for the investigational drug as a precipitant of CYP interactions in a screening capacity. If the AUC ratio is between 0.5 1.8 and 1.25, additional evaluation of the drug as a perpetrator is not needed. The working group discussed expanded utility of this approach, and the guidance indicates that when more relevant drug concentrations in the gut and liver can be determined, mechanistic static models may provide quantitative estimates of DDIs well beyond the current practices. The guidance indicates that PBPK models can assist in the evaluation of the DDI potential of a drug as an object or precipitant of enzyme or transporter-mediated interactions. When using PBPK modeling to support drug development decisions, it is essential to justify the following: the model assumptions, variability and uncertainty measures, and the physiological and biological plausibility. The guidance includes more details about the use of mechanistic static and PBPK models for evaluation of DDIs, and is open to additional utility as methods are further developed and assessed. As I indicated at the beginning of my presentation, the end of the DDI evaluation process should be useful information for the healthcare provider and patient. The risk assessment step translates the results of DDI studies into DDI management. In the guidance, DDI management is defined as DDI prevention and risk minimization. These strategies are needed when co-administration of drugs leads to concerns greater than when the drugs are administered alone. A few considerations when assessing the risk of a DDI include the variability of observed DDI data, the anticipated duration of concomitant use, the medical need for the drugs, including availability of alternatives, and the availability of monitoring parameters. Possible instructions for management include a change in dose level or frequency, staggered administration, prohibition of concomitant use, or monitoring drug concentrations, lab results, signs or symptoms, and possibly adjusting the dose. The guidance does not provide specific language regarding instructions for DDI management. However, as we consider the strategies, it is essential to consider them from the perspective of the healthcare provider and the patient. Can reasonable instructions be provided and followed? In conclusion, tremendous advancements have been made enhancing our understanding of DDIs. The collective effort from industry, academia, and regulatory agencies led to global harmonization. Over the past 5 years, there were countless in-person meetings, teleconferences that required early morning and late evening attendance from around the globe, and many hours of research, consultation, and writing. In spite of the amount of work, it was a tremendously rewarding process to participate in. The goal was clear to all participants. We wanted a scientifically rigorous process that keeps the patient population in mind. As you heard today, there are still areas that warrant further research to improve the efficiency of DDI assessments during drug development. Some of the areas include refining the extrapolation of DDI results, including in complex scenarios, incorporation of additional endogenous biomarkers, and defining further utility of predictive modeling, such as mechanistic static and PBPK. Before I close, I need to acknowledge the large group of individuals who contributed to developing the guidance. There were individuals from around the globe, from industry, and from other regulatory agencies, and there's a large number of individuals within FDA who contributed also. And many of these, including those in the audience, will also participate in the implementation of the guidance. Before we move to the next segment of the webinar, we will pause for 2 continuing education questions. Question number 1. What are the most important drug metabolizing enzymes? Carboxylesterases, cytochrome P450 enzymes, monoamine oxidase, or UDP glucuronylyl transferases? The answer to question number 1 is B, cytochrome P450 enzymes. Question number 2. A classification system for a drug as an inhibitor, strong, moderate, weak, or inducer, strong, moderate, weak, is available for which of the following drug interactions? Cytochrome P450 enzyme mediated, UGT mediated, drug transporter mediated, or all of the above? The answer to question number 2 is A, cytochrome P450 enzyme mediated. Thank you for your attention today, and I look forward to addressing your questions.
All right. Thank you again, Dr. Reynolds, for the excellent presentation. We'll now begin our next question and answer panel. And joining the panel, we'll also have Dr. Rajani Kant Matabushi. And for our audience, if you haven't, Dr. Matabushi is Associate Director of the Guidance and Policy Team within the Office of Clinical Pharmacology, within the Office of Translational Sciences in the FDA Center for Drug Evaluation and Research as well. So for our audience, if you haven't had a chance to enter your question into the Q&A chat pod, please do so now, and we'll answer as many questions as time allows. All right. So we can start with a question that actually can be addressed by Dr. Matabushi. What are the general recommendations or guidelines for demonstrating that we can reliably reproduce PPB binding greater than 99%?
Thank you, Nora, and greetings, everyone. It's an excellent question, and this was a topic of great discussion as people who have followed our draft guideline and its evolution to the final guideline. So specifically in anticipation of questions of this nature, we actually, in the guidance, included a specific section on plasma protein binding, or protein binding is what we call. It's the Appendix A, Section 7.2. The good practice considerations here are, when choosing a protein binding assay, it's important to ensure the suitability of the assay methodology for the investigational drug product. Second is, it's important to have a bioanalytical method that should have appropriate accuracy and precision at the required sensitivity ranges. For example, the calibration standards and quality controls at 15% level and the lower limit of quantification around 20%. Now, there have been questions also with respect to protein binding assay and its precision and accuracy and how to go about. And to this end, we have included several references which have at a very high level informed our thinking in this process that could be referred to. For example, if you're conducting a plasma protein binding study, you need to have positive controls to ensure in study performance, and the positive control should be demonstrated to be within threefold of the mean fraction unbound in plasma value that has been previously reported when the protein binding assay method was qualified. Similarly, when novel and emerging protein binding assay methods are being established, it would be useful to have the assay method qualified against an orthogonal method. And these good practice considerations are not uniquely developed by the ICH M12 expert working group, but they have relied heavily on the reported research and publications by experts in the field that have been out there.
Thank you so much for that response. We have another question for you. Does this guidance apply to small peptides?
Excellent question. So I think if you look at section 4.2, where we talk about therapeutic proteins and drug-drug interactions and the scope that we are addressing as part of this guideline, it clearly mentioned, along with peptides, other emerging modalities such as oligonucleotides, small interfering ribonucleic acids or siRNAs, modified ribose nucleic acids, and peptides are out of scope for this guidance. Having said that, back in December 2023, we from the FDA, we published a draft guideline on clinical pharmacology considerations for peptide drug products, and that guideline specifically articulates our FDA's thinking with respect to various clinical pharmacology considerations, and specifically among them are the considerations for assessing drug interactions. And there are sections that focus on pharmacokinetic interactions as well as pharmacodynamic interactions. So if people are interested, that would be a source to look out for. And when finalized, that would be implemented by the agency.
Right. Thank you so much for that response. And now we have a question that came in for Dr. Kellie Reynolds. Dr. Reynolds, if DDI is lower than for a weak inhibitor level, do we have to label?
Thank you for that question about labeling. That's an area where I work quite a bit. So the ICH guideline itself does not address labeling, and that's because it's a global guideline, so that would not be appropriate. And in general, for FDA clinical pharmacology guidances, we don't address labeling because usually there's a separate guidance that addresses labeling. But with that said, in general, for labels in the United States, the in vitro results are usually included in the labeling. But I think this question is referring to clinical study results where the result is less than weak inhibition. And the general principle for that is to include the information if it's clinically relevant. So I'm not going to say an absolute answer that we would never include the information, but typically we would not include that information in the label. We may have the results of the drug interaction study itself, but we wouldn't have a lot of information saying it's less than a weak inhibitor. Thank you for that question.
Thank you so much for that response. Another question that came in for you, Dr. Reynolds. What criteria should be considered and presented for validation are expected for, it looks like the abbreviation there is endogenous biomarkers?
Thank you for that question. Yes, there was quite a bit of discussion and interest in the endogenous biomarkers as we were drafting this guidance. And the performance characteristics that we would like to see for the assays are sensitivity, selectivity, specificity, the dynamic range, and then of particular importance, the correlation with pharmacokinetic parameters of probe drugs. And we would also like to see information about variability due to factors such as diet, age, exercise, diurnal variation, or disease state. Thank you.
All right. Thank you so much for that response as well. Well, we have another question that came in for you. Can nested studies be performed at steady state, for example, in oncology patients on daily regimens?
Thank you for that question. Again, nested DDIs was another topic that came up quite a bit during our discussions. And yes, they can be conducted at steady state, and typically they would be. As you see in the guidance, nested DDI studies, they are part of larger studies, so phase 2, phase 3 studies. So to be interpretable, for the most part, they would be conducted at steady state. And that is quite common for oncology drugs. Thank you.
All right, thanks so much. Now we'll do another question for Dr. Raj Matabushi. Are there any qualified endogenous biomarkers of 3A4 and UGT inhibition or induction, or at least that agencies would consider valuable?
Yeah, that's an excellent question, and there have been multiple questions that have come forth across specific enzymes as well as transporters that are listed. So if you look at our guidance where we talk in section 7, where we do talk about the considerations for a biomarker approach, there is a great interest for having reliable and sensitive endogenous biomarkers. If that was possible, it definitely would make our ability to characterize drug interaction potential in real time that much more effective. Having said that, not all of the biomarkers that have been reported out in literature have the adequate level of quality or consistency in findings to allow for reliable interpretation of results. And we do list several of these emerging areas where there have been potential for biomarkers across various drug metabolizing enzymes or transporters. And we do think that it would be useful for research to happen in this area where we can have biomarkers with data about their performance characteristics such as sensitivity, selectivity, specificity, dynamic range, all the attributes that Dr. Reynolds also mentioned, to be able to generate it for these to increase our confidence with respect to some of these biomarkers for areas that several of the audience are asking for questions. We did feel that we had a good level of information around the hepatic OATP 1B inhibition potential with the plasma proporphyrin proporphyrin 1 CP1 as an endogenous biomarker, and hopefully people will look at it and try to generate the relevant information to support some of the emerging biomarkers for other mechanisms here with respect to either metabolism or transporters.
Great. Thank you so much for that response. We'll do another question for Dr. Kellie Reynolds. Does this guidance apply to novel fixed dose combinations of 2 or more drug products, specifically if all the moieties are approved?
Thank you for that question. I recognize that it's not explicitly covered in the guidance. And so the principles outlined in the guidance apply to all drug development programs. So for a fixed dose combination, including a novel combination of 2 previously studied drugs, the in vitro information is likely already available. So whoever is developing the drug would need to think about what in vitro information are they already aware of? And based on that in vitro information, what is the anticipated net effect on other drugs, and what would be the expected effect of other drugs on the combination? And then the appropriate clinical studies could be conducted after that. And then the interpretation would be the same. So there would be some prior information already available, but the same principles do apply. Thank you.
All right, thank you for that response as well. Now we'll go back to some questions for Dr. Yang. Dr. Yang, is BSEP no longer in the initial transporter panel by EMA? And is it still a main transporter to evaluate for DILI with respect to bile acid homeostasis? Sorry, Dr. Yang, if you're speaking again, you might be on mute. There you are.
All right. Yeah, sorry. Yeah, I would like to clarify that the reason BSEP is removed from the final version is because BSEP, as you know, it's not much involved in drug transport. Relevance actually is about bile acid homeostasis, and the inhibition of BSEP could potentially block the excretion of bile acid into bile duct, and therefore potentially cause drug-induced liver toxicity. So because this is not really DDI, this is about drug toxicity profile evaluation. That's why we did not include it in the guideline. But that does not necessarily downplay the importance of that. I think this probably will depend on the individual company's strategy. This is part of the information could be used to evaluate the potential of drug toxicity on liver, along with other information. But of course, I think the audience also know that the DDA is quite complicated. There, could be the inhibition of BSEP alone doesn't necessarily cause DD, and on the other hand, DD can also be from other reasons than just the BSEP inhibition.
Great. Thank you for that response. We have another question for you, Dr. Yang. Can you further elaborate on the risk of false positive for intestinal DDI prediction using dose over 250 mLs? As you mentioned, many drugs have poor solubility, and their solubility in passive or effective is often much lower than dose 250 ml, and therefore can lead to false positive DDI prediction. What are your recommendations in these circumstances?
Yeah, that's a great question, which I know the answer. I think this issue has been there for quite a while. It definitely needs investigation from our community, industry colleagues and academic researchers to explore, see what might be the solution for this. I hope that in the future we could identify some endogenous biomarker for BCRP or PGP, just as what we found for OATP1B and several other transporters. Very recently, there has been some research publications, for example, by Dr. Hong Shen from BMS and from I think a Finland researcher group. They published a paper on the potential utility of riboflavin as an endogenous biomarker for BCRP. And also there's a poster presented at the ASCPT meeting early this year, from Dr. Cassie Giacomini group in UCSF, from a clinical study recently conducted. But of course, there's still much less the data in terms of this compared to several other transporters. Eventually, if we are lucky to have such an endogenous biomarker, I think that will greatly help improve the prediction and reduce false positive.
Thank you so much for that response. Another question came in for you, Dr. Yang. Could you confirm that CYP inhibition for metabolite greater than 25% of parent and less than 10% of radioactivity is not needed anymore?
Yeah, it's not needed anymore. But I would say actually the difference isn't actually not that. In the past, we published a paper collaborated with researchers from University of Washington, and we found that actually there's a good correlation between drugs with metabolite AUC compared to parent AUC more than 25%. Good correlation between that to the drug has radio activity accounted for more than 10% of radioactivity. That paper was published on clinical pharmacology and therapeutics in 2021. The corresponding author is Dr. Nina Iso-Harene from University of Washington. And also in that paper, we did a preliminary analysis showing that there's no clear relationship between the polarity of metabolite with the inhibition potency of a metabolite relative to the parent drug. I should have mentioned this reference in my slide. Sorry for that.
Thank you for that information. Another question that came in for you, Dr. Yang. The criteria of greater than six-fold induction for 2B6 PC may not be attainable for all lots. Should we look at greater than 20% PC when induction fold is less than six-fold for PC?
That's a good question. First of all, I want to clarify that this is a general expectation. But sometimes, let's say, we usually conduct this experiment using 3 donors. If 2 donors have the response to positive control more than six-fold, and with the other donor less than six-fold, it doesn't necessarily mean we have to add 1 more donor. We could still derive the same conclusion. In the when the positive control is less than six-fold, yeah, that 20% is used to avoid potential false negative prediction. I think this for CYP2B6, this actually may not be a big problem. I mean, for CYP1A2 and the CYP3A, because nowadays the hepatocyte we used usually, the positive control is six-fold. It's not an even problem for those at all. the issue still is mainly about the CYP2C enzyme. But as I mentioned in my presentation, we could leverage the in vivo induction effect for such an investigator drug on CYP3A. When the drug is found to be a potent inducer of CYP3A, meaning that it is a moderate inducer or strong inducer. We could leverage that information. Just to use a similar frame as very nicely presented by Dr. Reynolds in her presentation about how to evaluate and consider the drug as a PGP inducer or UGT inducer.
Thank you so much for that response. Now we'll turn to some questions that actually came in for Helen Heyman. So first question here, where can we find older versions of the ICH guidances online?
Oh, thank you for this great question, Nora. Yes, so one can find the older versions of guidances online. You simply need to type in fda.gov archive. Once you get to that landing page, what you will do is you will click on the date and look for a snapshot of the guidance that you're looking for so you can view it and download it. Thank you.
Thank you so much for providing that instruction. Another clarifying question you can maybe help us out with, is it M12 guideline or M12 guidance? Can we use it interchangeably?
Oh, yeah. Thank you for the insightful question. It really depends from what stage and what perspective you're speaking. So, this is perhaps how to look at it. As I was showing in my presentation, the ICH guideline is the result of a successful collaboration between regulatory agencies and industry organizations under the auspices of the ICH. The FDA guidance is then the adopted ICH guideline by the agency in August 2024. And both do talk about principles and recommendations that it intends to follow. Thank you.
Thank you so much for that response. Now we have some questions that came in for Dr. Kellie Reynolds. If strong, moderate, weak category works only for CYP enzymes, what categories are assigned for UGT and PGP mediators?
Thank you for the question, and I recognize that the frustration of not having categories. Right now, we only have categories for the CYP, so the categories that were mentioned in the question: the weak, moderate, and strong. And there are no categories for any other enzyme systems or for transporters. I know there's been particular interest in categories for PGP, and the reason we don't have the categories for the other systems is because for most of the drugs that are involved with interactions by those pathways, there are other transporters or enzymes involved in their elimination, and it makes it very difficult to extrapolate and get the full utility of the categorization system. We're still interested in coming up with something, but at this point, we do not have something that can be confidently recommended. Thanks.
Thank you so much for that response. Another question for you, Dr. Reynolds. What are the criteria to conduct or develop the study in healthy volunteers and in patient or disease patient?
Thank you for that question. Most drug interaction studies are conducted in healthy volunteers, and that's because it's just a cleaner assessment. It's easier to conduct as a phase 1 study, and we're assuming that the results could be extrapolated to the patient population. Now, if there's safety concerns and the drugs involved in the drug interaction study cannot be given to healthy volunteers, certainly the study would be done in patients. It is also acceptable to conduct any drug interaction study in patients if it can be interpreted. So if there's nothing about conducting the study in patients that would make it difficult to interpret, then the study can be conducted in patients. In particular, the nested DDI studies are all conducted in patients. So there are not specific criteria saying you must do it in one population or the other, except for the fact that if it's not safe to give to healthy volunteers, it should not be done in healthy volunteers. Thank you for that question.
Right, and thank you so much once again for that response. And now we'll go to a question for Dr. Yang. If the drug candidate has high passive permeability and is mainly cleared by metabolism, is there a need to evaluate its potential to be a substrate of hepatic drug transporters such as OATP1B1 or OATP1B3?
Yeah, that's a great question. I think the audience also noticed that there's some changes from the previous guidance, where we say that the the risk, for drug as substrate OATP1B is many ways drug with low solid, low permeability. This this is removed, but there are some nuances here. It's because depending on how we define the low permeability or high permeability, there are some publication in literature, such as from Dr. Warma and his colleagues in Pfizer, and they demonstrated that for some drugs which they propose using a ECCS classification system, I think it's a ECCS 1B category. It's a ionic compound with a large molecular weight more than 400 Dalton or 430 Dalton. For some of those, even though they show the reasonable permeability, in vivo they may still subject to the impact of OATP1B inhibition. But I would think this really depends on how this permeability, in vitro permeability, is categorized. I think this probably more fall into the middle area, the gray area. For drug with quite a high permeability, I would expect the transporter, including OATP1B, play a limited role. And for drug with clear lows of permeability, of course, we expect that transporter may be important, but when it fall into this middle, there's some challenge. So in the guidance, we remove this, but it's not saying that we do not acknowledge the importance for permeability in the overall assessment.
Right. Thank you so much for that response. We have another question for you, Dr. Yang. Recent publications support use of carbamazepine with a shorter induction duration than what was previously used. Is there any guidance on the recommended study design for a carbamazepine CYP inducer study?
Yeah, that's a great question. First of all, in the guidance, we have a paragraph, talk about the study design consideration for inducer drug. That's a general principle. So it has come to the individual inducer drug. Use carbamazepine as an example. In the past several years ago, when we provided the recommendation to companies. At that time, there was very few clinical DDI study with index substrate of CYP3A. I think there's only one study around that time published with midazolam. And after that, there are a few new clinical DDI study conducted, which greatly enhance our knowledge on this. For example, a recent study presented at the ASCVT annual meeting last year. It's very nicely that the company compared the shorter pre-treatment duration of carbamazepine and show that I think around 10 days pre-treatment duration, it showed similar effect as a longer pre-treatment duration. So I think this provides direct clinical evidence supporting the shortening of the overall treatment period, which could help reduce the happening of adverse event, right? In general, we think, the longer treatment, the more likely adverse event happening. I think this is an excellent example. The the further investigation from the researchers in our community, including industry colleague, to enhance our understanding, and which could update our recommendation or the consideration when design protocol.
All right, thank you. And we have another question for you, Dr. Yang. How does FDA view in vivo non-clinical studies and their utilities?
That's a very interesting question. I think this we don't often use this. It's mainly because of the difference and the potential difference between species, animals versus humans. So usually we start with human-related material in vitro, and then predict in vivo. Having said that, I know that there are some research efforts to develop transgenic animal model, like a transgenic mice, knockout, knock-in, and also there are some studies using like dog, depending on the purpose of assessment. I think there's some utility of like a proof of concept, and this probably will be case by case. It will be part of totality of the evaluation package, and also the cyno monkeys. I think people also use that for OETP1B inhibition prediction, because cyno monkeys seem to have a great similarity in terms of OETP1B with humans.
Great, thank you. And now we'll have a question for Dr. Kellie Reynolds. Can you update the DDI examples page on FDA with new information?
Yeah, and thank you for that question. We know that the examples it's quite a popular web page, and yes, we can update them periodically. We do update it with new information, either from new literature that we become aware of, or because new drugs are approved. So it is something that we look at on a regular basis, and we can update the information. Thank you.
Great, thank you so much for that response. We might be able to fit in one more question. This will be for Dr. Raj Mutabashi. How can we apply the guideline concepts to biological products?
Yeah, thank you very much. And as mentioned in the guidance, clearly for the biologics or therapeutic proteins, the guidance is very briefly touching upon the range of drug interactions. And in fact, the approach here is to understand the effects on the small molecules, which is where most of the hepatic metabolism and transporter effects are well understood. So from that perspective, there are several mechanisms that are listed. Certainly, there are some therapeutic proteins that may exert an indirect effect on the expression of CYP enzymes, and thus affecting the pharmacokinetics of small molecules, and that is of consideration. To this end, we also have an independent guidance from the FDA, which was published in June of 2023. This is a final guidance on drug-drug interaction assessment for therapeutic proteins, and that provides a lot more details associated with the drug interaction potential for various therapeutic proteins. Analogous to this is also another concept on the antibody drug conjugates, which are also a class of biologics for antibody drug conjugates, where there is a small molecule drug component that is conjugated to the antibody. And from the drug interaction potential perspective, though we would be interested in both antibody and small molecule, for this particular guidance, the focus is predominantly on the small molecule aspect, and it is important to understand the formation, distribution, and elimination kinetics of the small molecule that would be part of this ADC, and it would be important to evaluate all the aspects associated for that small molecule component as a substrate drug. Hopefully that helps address the questions around biologics as well as ADCs.
Yes, thank you so much for that response. So that looks like that's all the questions we have time for with this session. Thank you again to all our presenters and panelists for the excellent information.